Hello and welcome everyone to this podcast for cell biology. In this podcast we're going to talk about something called exocytosis, which we often refer to as secretion, and we're also going to talk about endocytosis and a few other things linked to endocytosis. And when we talk about endocytosis, we're going to talk about it from the perspective of recycling. How do we recycle some of the molecules involved in cellular events? Okay, as we begin this process, let's draw a simple model up here outlining secretion and endocytosis. And I'm going to begin with the nucleus here. This is just part of the nucleus, and I'm going to, let me label that first, nucleus. And I'm going to remind you that the nucleus is continuous with the ER. I'm going to just draw the ER like this, connected to the nuclear membranes. And let's label this ER. Next, let's draw the Golgi apparatus. And the Golgi has several fairly large membrane structures, like so. We call the one closest to the ER the cis Golgi, and the one further away we call the trans Golgi. But they're all Golgi apparatuses. So we're going to have a protein, and we're going to talk a lot about this in just a moment, but a protein that begins to be made in the cytosol, but before it finishes, it enters the ER. And now we have an ER protein. This ER protein we'll talk about in a little bit could either be in the lumen or it could be on the membrane. We'll go ahead and draw two of them. One that remains in the membrane, one that's in the cytosol. Now ultimately, remember, we want to get this protein all the way to the outside of the cell. So this is our plasma membrane, just abbreviated as PM. But we want to get that protein out here. So how are you going to get this protein that is in the ER all the way over here? What happens initially is a vesicle fuses from here. Inside this vesicle are going to be the ER proteins. You may have an ER protein that was in the lumen, or you may have one that's in the membrane, but it just pinches off of this ER. So remember, this membrane here was once part of the ER membrane, and it contains the protein that we want to move. Now this ER protein in this vesicle, in fact, why don't we write vesicle here? This vesicle is going to move, and eventually it's going to fuse with this Golgi. And when it does, it will release the lumen protein that was here originally, and then it will also transfer from the membrane of this vesicle, the transmembrane membrane protein, it'll transfer to the membrane of the Golgi. Now these proteins, they may be proteins that need to stay in this Golgi, but for now we're going to talk about proteins that need to get out of the cell. So these proteins can't just move through these stacks by themselves. They must form new vesicles, just like we saw here. A vesicle is going to pinch off of this cis Golgi, and it's going to move to this medial Golgi. And then a vesicle is going to pinch off this medial and now join to the trans Golgi. And so at the end here, you will have your membrane protein and your luminal protein in this trans Golgi. Now ultimately, as we've said before, the goal is to get this protein to the plasma membrane. And so once again, not surprising, these pinch off into a vesicle. And this vesicle has, again, that membrane protein, or maybe has this lumen protein. This vesicle now will travel to the plasma membrane, and at the plasma membrane, it will fuse with the plasma membrane, and in that process, it will release the lumen protein to the outside of the cell, and it will insert this transmembrane protein into the membrane of the plasma membrane. So this whole process, from the protein entering the ER to the vesicle to the Golgi to leaving the Golgi to entering the plasma membrane, we call this secretion, or we call it exocytosis. Now the reverse, endocytosis, let's write that up here, endocytosis. This is when we have a protein or some other molecule on the outside of the cell, and we want to bring it into the cell so it can be digested. And so by various mechanisms, and we'll talk about these towards the end of this podcast, this molecule will fuse with this membrane, and when it fuses with that membrane, it will form a vesicle. This vesicle we call the early endosome. And it's going to contain this molecule that we just brought into the cell. Now this late endosome is going to fuse with another vesicle, and this vesicle has pinched off this Golgi. It will have a set of proteins in it as well that are going to be important in transforming this early endosome into a late endosome. Late endosome. This late endosome, now it contains this molecule that was brought into the cell from outside. It will contain that molecule. It will also contain enzymes brought to it from this vesicle from the Golgi. And when they fuse, they form, as I said, this late endosome. This late endosome will either fuse with a lysosome, or more likely, it's going to mature into a lysosome and degrade this molecule that was brought into this cell. And so I'm just going to draw a couple dots here, indicating that it's been broken up. And so that's what we mean when we say it recycles things. It breaks these larger molecules up into smaller, more usable components that the cell can use. So I want to spend just a moment here talking about these vesicles. There are two features of these vesicles that must be true. The first is that it must have the correct cargo. And when I say cargo, I mean the protein. Either this protein here, this protein in this vesicle, the protein that's being brought in from the outside. It has to have the correct cargo. And the second thing is that these vesicles have to fuse with the correct target. Now, how they accomplish these things we'll talk about later on, but you will have to learn some of this in greater detail in more advanced courses. But it's important for you to know now that these vesicles are unique. They have the correct cargo and they have the correct destination tags on it or proteins on it to send it to the correct locations. So we don't have ER vesicles fusing directly with the plasma membrane. Well, as we look at this process again, thinking about how that protein moves from the ER all the way to the outside of the cell, it's important to remember that, of course, we did not always know this is how a protein made it from the inside of the cell to the outside of the cell, until a scientist by the name of George Palade took up this question, and he used this technique called pulse chase. And we'll talk about this in just a moment. Now, he could have picked many different kinds of cells. Remember, we talked about in class, picking the right cell type or the right organism is important. Now, the one he picked may have been his best choice. He chose a pancreatic cell because the pancreatic cell is known to produce a lot of enzymes and secrete these enzymes, more specifically digestive enzymes. It's one of its main jobs. It just keeps producing these enzymes over and over again. So if you're going to study secretion, start with the right cell type, and that's exactly what he did. Now they knew that these digestive enzymes, I'm just going to write DE, were produced and taken out of the cells, but they didn't know how. They didn't know if it was just made into cytosol and then exported out, or if it had to go through some kind of processing, going through some kinds of organelles before it could leave the cell. They just didn't know. Now you can't just, under a microscope, without any aid, see these digestive enzymes moving within the cell and out of the cell. But if you could somehow label these digestive enzymes so that you could see them as they go throughout the cell and are excreted or secreted outside of the cell, then you might be able to discover the pathway. And so that's when we come to this pulse chase experiment. The first part of the experiment is to allow these cells to grow normally. So we can maybe write that down here. And we're going to call this before experiment. Cells would just grow. Then this next part of the experiment, this was the pulse. And this is the part where we're going to label the protein. And the way that the proteins were labeled was that they put in, I'm going to draw it over here, radioactive leucine. Just like the leucine we learned about earlier in the semester, the amino acid. And they placed that in this cell during this pulse part of the experiment. During this pulse part of the experiment where they're allowing this leucine, this radioactive leucine to come in here, that means all proteins made, all these digestive enzymes that are made, are going to be radioactive. So radioactive proteins. Now this part of the experiment is relatively short, only about 3 minutes, which is enough time to incorporate that radioactivity. Then this next part of the experiment is called the chase. What's happening during this part is that we're watching these enzymes that were just made with radioactivity. We're going to watch these proteins, or we're going to chase those proteins through the cell until they exit the cell. So chase the radioactive proteins throughout the cell. So what they would do is at various time points, your book says 7 minutes, 17, 37, 57, and then 117 minutes. They would take cells that were growing in a flask and figure out what part of the cell the protein was. They also looked at a 0 time point. That's an important one. And so what they did was, so at 0 minutes, and we're going to use the diagram up here to talk about this, at 0 minutes, the protein that was labeled is going to be in the ER. Now remember, all other proteins that are going to be made after this initial 3 minutes won't have any radioactivity. So it's only these proteins made in this first 3 minutes, only those proteins will be radioactive. So at 0 minutes, all the radioactivity was in the rough ER. Okay, they let the time go a little bit longer, and they looked at 7 minutes. And at 7 minutes, what they discovered was about half of it was in the rough ER, and about half of it was in the Golgi. And then as you move to 17 and 37 minutes, we started seeing more and more of that radioactive protein in the Golgi. You'd still have some in the ER, but as that protein was being processed through the ER, as time went on to 37 minutes, most of that protein ended up in the Golgi. And so let's erase this from the ER for now and indicate that we have these proteins now predominating in the Golgi. Though we can't see it, it's important to remember more proteins being taken into the ER. It's just not radioactively labeled, so we can't detect it. We're only detecting that protein that was made during that initial pulse time for 3 minutes. Now, as time went on a little bit more, say at 57 minutes, they saw fewer and fewer proteins in the Golgi, but started to see more and more of this radioactive protein in these transport vesicles as they're moving towards the plasma membrane. So vesicles at 57 minutes are now radioactively labeled. At this 57 mark here, you'll still see some Golgi with radioactively labeled proteins in it, but it's getting much less. The predominant is going to be these vesicles. Then at 117 minutes, essentially all of that radioactivity is going to be found in the vesicles. And we're going to start to see some of that radioactivity right here at the membrane, because it's getting ready to fuse with this plasma membrane and to be released. After 117 minutes, we're going to see more and more, so I'm going to say 117 minutes plus. We're going to see most of that, I forgot to put plasma membrane, sorry, let's go back here and do that. Alright, so at 117 minutes, you're going to see most of that radioactivity at the plasma membrane plus released from the cell, secreted from that cell, so it's outside here. That radioactivity is now outside of the cell. So again, I'm going to walk through this one more time. This experiment is done by taking your cells and radioactively labeling the proteins for 3 minutes. As the proteins are being made, they're going to enter the ER, the rough ER. At the rough ER, early in this time period, around 7 minutes, most of the radioactivity is going to be found in this ER. It then begins to accumulate in the Golgi. That is, the radioactively labeled proteins will accumulate in the Golgi. As the time goes on, less and less radioactivity is found in the Golgi, and more are found exclusively in these transport vesicles. And then finally, at the end, we will see more and more of that radioactively labeled protein being secreted from the cell. And that this last several parts here, that was remember, the chase part. Following that radioactivity as it goes throughout the cell. Okay, so that's how they first determine the pathway of secretion, or exocytosis. Now I want to go back to the beginning here and think about how this protein first enters the ER and then later leaves as a vesicle. Then we're going to look specifically at these other downstream effects too. But let's first look at how the protein enters the ER. So what I'd like to do then is take this area here and expand it down here. I'd like to keep this up here throughout the rest of the podcast just so we have a visual of where everything's at. So let's draw our ER down here, like so. And let's think of this whole space here, down here, as our inside of this cell. Inside this cell, we're going to have ribosomes that are going to take messenger RNA and begin to synthesize a protein. As it begins to synthesize this protein, at this end here, which is the N-terminal end, there is this targeting signal. This targeting signal is what's important to tell this protein it needs to go to this ER. Now the targeting signal by itself can't get to the ER, but rather what happens is another protein comes along here and binds it. And this other protein is called SRP. I'm going to draw over here. SRP stands for Signal Recognition Particle. It recognizes this ER signal, which is a specific sequence of amino acids. It binds this and brings it to the ER. The ER has a protein here, and this protein here that's a transmembrane protein, it's called a SRP receptor. And I don't have room, so I'm just going to write REC. But this protein, this RSP receptor, will bind to this SRP. And when it binds to this SRP, it brings it to this membrane. So in the ER, the protein folds. Now, as the protein folds in here, this is something I didn't tell you. I'm just going to draw some C's in here. There are proteins called chaperones. These are proteins that reside in the ER, as well as the cytosol and other organelles, but we're just talking about the ER now. So these chaperones are in the ER lumen, and they will assist in the proper folding of this protein. So chaperones in ER. Now the last thing I want to say is that ER proteins become glycosylated, meaning they become glycoproteins. And as they reside in the ER, they can become more and more glycosylated. And as we'll talk in a moment, we can determine the maturity of the protein by how much it has been glycosylated. Because as we go through secretion, it will pick up more and more sugar residues on the outside of that protein. Now remember, our next step is the Golgi. I'm going to just redraw that down here. Our different Golgi stacks. Remember, we go from cis to trans. As we mentioned before, the way that this protein gets from the ER to these Golgi apparatus is it buds off from the ER to form a vesicle. And this vesicle will now contain our fully folded ER protein. It will now fuse with the cis membrane of the Golgi. And as it fuses with it, this ER protein will now be placed in the Golgi. So we should just call it a Golgi protein now. Now what happens here, this protein will now be packaged into a vesicle from the Golgi that contains this protein. It will fuse with this middle Golgi, then it will be released from that middle Golgi, and then once again it will be fused to the next one here. Now I didn't draw it, but remember that at each of these stages, this protein resides in each of these Golgi membrane structures for a period of time. So what's happening in the Golgi as we move in this direction? Well, a few things I want to remind you is I only drew 3 Golgi stacks here, but we know there can be thousands of these Golgi stacks, and these proteins will move between them. And as they're moving between them, the proteins in the Golgi are further glycosylated and processed. It's kind of a finishing area for these proteins. So at this point, we talked about glycosylation in the ER and in the Golgi. I just want to talk a little bit about the importance of this. Remember, glycosylation is adding sugars to these proteins. This glycosylation can protect proteins from degradation. Those sugar residues on them, on these proteins, block the enzymes that often will degrade proteins. Holds protein in place. And what I mean by that is the timing of the protein moving from the ER to the Golgi to the different stacks of the Golgi is precisely timed. And the one way it is timed is by the degree of glycosylation that occurs. Earlier steps have fewer sugar groups added onto it. Later steps have more sugars added onto it. They can be transport signals. When they're on the outside of membranes within the cell, they can indicate the proper destination of certain vesicles within the cell. And we mentioned this last one before, but they identify self cells on the plasma membrane. So if they reach the plasma membrane, these glycosylated proteins may tell the immune system that these are our cells. Don't kill these cells. Okay, so now, as this final vesicle buds from this Golgi, it has one of several destinations it could go to, some of which were hinted above. So first off, this vesicle could stay in the Golgi. It may return to the Golgi. It may go to the different areas of the Golgi. It could go to the lysosome or fuse with an endosome to eventually become a lysosome. It could also complete the secretory pathway or exocytosis, and that protein could leave the cell, if this is our plasma membrane, it could leave the cell releasing our good friend, this protein, which started way back here. These are the three main pathways. It can stay in a Golgi or go back to the ER, or it could go to a lysosome or fuse with an endosome, which would then eventually become a lysosome, or it can be secreted outside of the cell. Alright, now I want to spend the rest of our time talking about recycling at the cellular level. So either the cell will bring in material into the cell to fuse with the lysosome and to be degraded, and then those smaller components of that molecule that was degraded can be used by the cell. Sometimes there are parts in this cell, could be organelles, that need to be degraded, and so they will also go to the lysosome to be degraded, and so that the parts that are being degraded can be reused. In many ways, our cells are incredibly useful at recycling material. We can think of 3 different ways that a cell can recycle larger molecules, larger materials, into smaller usable materials. They include receptor-mediated endocytosis. They also include phagocytosis. And finally, they include autophagy. I'm going to spend a little bit of time talking about these, but these first two, phagocytosis and receptor-mediated endocytosis, are examples of endocytosis, which simply means you're bringing something from outside of the cell into the cell. Autophagy, which we won't be able to talk too much about, can be think of as cellular eating, or more specifically, it digests old organelles and other large structures within the cell. It doesn't bring it from outside the cell. It begins to digest material within the cell. So let's go ahead and talk about these 3, particularly the first 2. And I'm going to begin with receptor-mediated endocytosis. In this particular example, here's our cell. On the outside of the cell, there are going to be receptors. There's all kinds of receptors, and they have different kinds of specificity to different kinds of molecules. But let's say these receptors bind specifically to this molecule that's on the outside. And whatever it is, it wants to bring it inside the cell so it can be degraded. These receptors, just like enzyme active sites that we talked about before, have a high degree of specificity to these specific substrates. They're going to bind into these receptors, and as they do, what happens is this membrane begins to pinch inside of the cell. Something like this. Still with those receptors on it, and still with those substrates bound to it. And then at a later time point, what happens is this pinched in part of the membrane will fully form into a vesicle, sealing back up the cell, of course. But within the cell now, you have an early endosome. Again, because these substrates had specificity with these receptors. It brings it into the cell. This early endosome, if you remember up here, will become a late endosome when it fuses with a vesicle from the Golgi, like this, and then that can mature into. In that way, these substrates that were on the outside of the cell are now going to be degraded by that lysosome. And just real quick, we won't go into great details, but if you're wondering how this early endosome turns into a late endosome and eventually matures into a lysosome, remember what we mentioned before, is that from the Golgi we're going to get these vesicles that are formed, and it's going to contain digestive enzymes that are going to digest any material it comes in contact with. And as it fuses with this early endosome, it's going to dump in those enzymes that are going to degrade this substrate, or this substrate down here. But it takes a while for it to become active, because remember, earlier we talked about the low pH of the late endosome. It takes a while for this pH of the late endosome to drop low enough before those enzymes are active. And when they become active, they will begin to degrade that substrate brought in. Okay, so let's talk about phagocytosis. Phagocytosis is similar to what we talked about, receptor-mediated endocytosis. It's not quite as specific, but it takes in much larger molecules. So if this is our cell here, and this is the larger structure it's going to take in, maybe it's a bacterium, maybe it's something else. But what happens is, as it gets closer here, the membrane will open up and wrap around this larger structure here. It will continue to engulf this outside structure until it's all the way inside, and then these membranes will seal off like so. This becomes a structure that we call a phagosome. Unlike receptor-mediated endocytosis, where the early endosome becomes ultimately the lysosome, the phagosome will fuse with the lysosome. When it fuses with the lysosome will take in this large particle and chop it up into little tiny pieces. All of those pieces, those molecules that made up this large molecule, are now recycled throughout the cell in many different ways to build other new larger structures. Let's spend just a little bit talking about autophagy. As I said earlier, autophagy is considered cellular eating. It's when the cell begins to eat parts of its cell. In this example here, we have our lysosome, and we also have all these kinds of organelles within the cell. Let's just draw a mitochondrion here. Now over time, this mitochondrion will age and become less functional. In fact, it could become dangerous to the cell. And so what happens is, these old organelles will be taken up into the lysosome. And in the lysosome, they'll be chopped up and digested. So it removes old organelles. You should look at receptor-mediated endocytosis, phagocytosis, and autophagy and think about and compare the similarities versus the differences. Okay, that ends this podcast here. And just as a quick overview, we talked about the many different steps in exocytosis, taking a protein that's made in the cell through the endomembrane system, that is through the ER, these vesicles, the Golgi, and then the plasma membrane and outside the cell. It also includes getting things to the endosomes and ultimately the lysosome. We also then spent some time talking about endocytosis, or how we recycle molecules, either from the outside, where we can use receptor-mediated endocytosis or phagocytosis, or it's a way we can recycle old organelles through autophagy. That's all I have for this podcast, but if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.